Good morning, dear students of class 12 science. Welcome to biology class. We are dealing with the unit number 4, biotechnology and its applications, and we have started with the chapter number 11, biotechnology, principles and processes. In the last class, we have started with the definitions of biotechnology, which has been defined by various international organizations like OECD. Then we have got US National Science Federations, and the most important one, the European Federation of Biotechnology, EFB. We have also discussed about the history of biotechnology, where we have learned about the phases of biotechnology. One was the traditional biotechnology or classical biotechnology, also known as the old one. Then second phase is the modern biotechnology. Then we have discussed about the principles of biotechnology, where we have got two techniques. One is genetic engineering, also called recombinant DNA technology, and number two is biochemical engineering processes. Under genetic engineering, we have learned about the formations of recombinant DNA technology, because Herbert Boyer and Stanley Cohen, they were the first to produce the recombinant DNA. Okay, so this till here we have discussed in the first class. Okay, so today we will continue with the same topic, that is recombinant DNA only. So first of all, we will just see the discovery of recombinant DNA technology. All right, the technology that lead to the formation of recombinant DNA, where the DNA will be from two different sources. Okay, so how this technology came into existence, how was it discovered, and what were the possible possibilities that lead to the discovery of this technology. So first one, we can say that this technology came into existence, evolved mainly due to the certain features, certain characteristics. Okay, the first features is due to the denaturations and renaturation of DNA. Now what do you mean by denaturation and renaturation of DNA? Now try to recall that what you have learned in class 10 and in class 11. We all know that DNA is a double-stranded molecule, like two strands. Clear? And within the DNA, we will find different type of base pair. We have got adenine, thymine, guanine, and cytosine. If you remember, ATGC, right? A will pair with T, and G will pair with C. Okay. So in one strand, if there is A, and in another strand, if there is T, so they will pair up, and they will form a double-stranded DNA. So what does denaturation of DNA means? Listen very, very carefully. Okay. Denaturation means under laboratory conditions, okay, under lab conditions, when we used to heat the DNA up to 100 degrees Celsius, 90 to 100 degrees Celsius, then the nitrogenous bases between the DNA, they will break up. Okay, they will break up, and the double stranded, if they will break up, then they will form a single stranded DNA. Okay, so whenever the double stranded DNA will separate and form a single strand, that is called denaturation. Within our body, we have got different type of enzymes which are involved in the denaturation of DNA. But I am saying that when in lab conditions, when we used to heat the DNA, then the hydrogen bases between the DNA, between the nitrogenous bases of DNA, they will break up and will make the DNA single stranded. That is called denaturation. Alright, now what is renaturation? So on cooling, when the temperature keeps on decreasing, alright, on cooling, the complementary strands of DNA again, they will pair up. Clear? The complementary strands of DNA again, they will pair up. If in one strand there is adenine, and in the other one if there is thymine, then they will pair up. If there is guanine on one strand and cytosine on other strand, they will pair up, and again they will form double-stranded DNA. That is called renaturation. And this pairing will be according to the complementary base pairing rule, where adenine will pair with thymine and guanine will pair with cytosine. So this is called denaturation and renaturation of DNA. Clear? Number two possibility is due to the in vitro or artificial synthesis of gene, having the gene of interest. Okay, where we can create the multiple copies of a gene having the measurable characteristics or the segment of DNA. Number 3 is the discovery of restriction endonuclease enzyme. If you remember, it is called the molecular seizure. Okay, then use of plasmids or phase DNA as vector. Vectors means where we will just put the gene of interest. Alright, so these are the 4 possibilities that leads to the discovery of recombinant DNA technology. Now just briefly I will try to recall you what we have learned in our previous classes. Okay, so in our first class we have learned that within the foreign DNA we have got a gene of interest. Gene of interest means having the desirable characteristics. Okay, so that is present in the foreign DNA. So only a segment of DNA has that characteristics which we want, which is desirable. Then we require a vector. So in this case we are using the plasmid vector. Okay, now what is the role of vector? Vector will help to carry, will help to replicate or multiply the segment of DNA, the foreign DNA having the gene of interest. Okay, so what we do now we have got the foreign DNA where we have got the gene of interest, and we have got the vector DNA, that is plasmid vector. Now we will require the use of molecular seizure, and that is restriction enzyme or restriction endonuclease. Okay, that which we have done in the previous classes. This is just a recall for you. Okay, so what we do, the same restriction enzymes will be utilized to make a cut on the foreign DNA on the plasmid vector. Clear? Okay, so by this way we have created a segment of DNA having the gene of interest, and similarly we have made a cut on the plasmid vector also. Okay, so what we do next, now we will ligate or we will join this segment of DNA having the gene of interest into the plasmid vector. Remember, the same enzyme has to be utilized to cut the foreign DNA and the plasmid vector. Clear? We will not use the 2 types of restriction enzymes. The same enzymes will be utilized to make a cut on the foreign DNA and on the plasmid vector. Okay, so now these particular ligations, this particular joining of foreign DNA having a segment of gene of interest into the plasmid vector is possible due to the discovery of molecular glue, that is the enzyme known as DNA ligase. Okay, so the enzyme DNA ligase are mainly utilized to join the foreign DNA to the plasmid vector. So finally a new DNA will be produced having the DNA from the foreign source, from the foreign DNA, and the DNA from the plasmid DNA. Okay, so this type of DNA is known as recombinant DNA because it has the DNA from 2 different sources, one from the foreign DNA and another one from the plasmid DNA. So this DNA is known as recombinant DNA. After the formation of recombinant DNA, it is introduced into the E. coli cells. Alright, then we require the next enzyme, polymerase enzyme, that will help in the replication of this recombinant DNA, because the polymerase enzymes of E. coli, Escherichia coli, the bacteria, okay, will recognize the recombinant DNA as its own and will lead to its replication or multiplications, where it will keep on dividing. So one cell will divide into 2 daughter cells, 2 into 4, and hence it will keep on dividing, and as a result we will be able to get the multiple copies of recombinant DNA. So these things we have learned in our previous classes. So therefore, by the discovery of the DNA ligase enzymes, by the discovery of restriction enzymes, by the property of denaturation and renaturation of DNA, and due to the artificial synthesis of gene, it was possible for the discovery of recombinant DNA technology. Okay, so I have mentioned about the denaturations and renaturation of DNA. Okay, now we will talk about the tools of recombinant DNA technology. Clear? So kindly go through the discovery of recombinant DNA technology from textbook. Okay, page number 580, and the diagram figure number 11.1, it's in the page number 581. Clear? So now we will discuss about the tools which are being used for the recombinant DNA technology. Okay, we have got different types of tools which have been employed in the field of biotechnology, in the field of recombinant DNA technology, and those tools are mainly enzymes. Okay, first enzymes, if you remember, we are using the enzymes restriction endonuclease, we are using the enzyme DNA polymerase, we are using the enzyme DNA ligase. Okay, so just want to remember that different tools are required for the formation of recombinant DNA by the help of recombinant DNA technology, and the first one is enzymes. Okay, second one, cloning vector. Now you can tell me which vector we are using. Plasmid vector, though we have got different types of vectors, we'll discuss afterwards. Clear? Then we have got foreign DNA having the gene of interest, and we got the competent host. Okay, so these are the different tools of recombinant DNA technology: enzymes, cloning vectors, foreign DNA, and the competent host. Okay, so for today we will just learn about the enzymes. Clear? In our third class of this chapter we will discuss about the other tools, but today we will simply focus on the enzymes only. Clear? So let us begin with the first tools of recombinant DNA technology, that is the enzymes. And following types of enzymes are needed in recombinant DNA technology. The first enzyme is lysing enzymes, and the second enzyme is cleaving enzymes. Now what do you mean by lysing enzymes? Okay, L-Y-S-I-N-G. Lysing enzymes means these enzymes are mainly used to open the cell or to dissolve the cell wall of plant cells. Now the plant cell wall is made up of what? Cellulose, we can say that. Okay, so in this case what we are doing, we are using the enzyme lysozymes, L-Y-S-O-Z-Y-M-E-S. So we are using the enzyme lysozymes, which are used to dissolve the bacterial cell wall. Listen very carefully. Okay, so in lysing enzymes, these enzymes are used to open the cells or to dissolve the cell wall of plant cells. Then again we are using the enzyme lysozymes, which are used to dissolve the bacterial cell wall. Clear? Okay. Number 2 is cleaving enzymes, which are used to clip the DNA molecule into segment. So it will keep the it will clip, all right, it will clip, it will join, or it will attach to the segment of DNA in simple term. Okay, it will attach to the nitrogenous bases of the DNA molecule. Nitrogenous bases means adenine, thymine, guanine, and cytosine. So it will recognize those base sequences and it will clip on them. Or to open a circular plasmid DNA, if you have seen the first video, first diagram 11.1, you have seen that we have got the vector DNA having a circular DNA molecules which has been cut. So that cut is mainly done by the help of the cleaving enzymes. Okay, so these enzymes are called nucleases, and nucleases are of three types: exonuclease, endonuclease, and restriction endonuclease. Okay, so lysing enzymes are mainly used to open the cell of plant cells, and lysozymes are mainly employed to open the cell wall of bacteria. Then cleaving enzymes, which are used to clip the DNA molecule into segment. Alright, which you should remember, the foreign DNA has been cut by the enzymes to just take out the gene of interest. Then the same enzyme was used to open the circular DNA molecule also, plasmid DNA also. Okay, so these enzymes are called nucleases, and which are of three types: exonuclease, endonuclease, and restriction endonucleases. So they are called nucleases because they are mainly used to clip the DNA molecule. So let's see the first one, exonuclease. So exonuclease activity, okay, it removes the nucleotides from the 5' end or 3' end of DNA molecules. Now what do you mean by nucleotides? What is nucleotides? So before going nucleotides, try to recall the structure of DNA molecule. Okay, if you remember, we have got nucleosides also. We have got sugar. We have got nitrogenous bases, adenine, thymine, guanine, and cytosines. Then we have got most important one, phosphate group. So sugar plus base plus phosphate will form a nucleotide. Clear? Sugar and base will form nucleoside, N-U-C-L-E-O-S-I-D-E. But nucleoside plus phosphate will form the nucleotides. Now here you can get confused because I am saying nucleoside plus phosphate. Why I am saying nucleoside? Because nucleoside means sugar and base, and there are 4 types of bases we can say: nitrogenous bases, adenine, thymine, guanine, and cytosine. Clear? So this exonuclease will remove the nucleotides from the 5' end or from the 3' end of the DNA molecule. Look at the diagram 11.2. So you can see over here the nucleotides are being removed from the 5' end of the DNA molecule, and this is mainly due to the activity of which enzyme? Exonuclease enzyme. Exo means outside, from the end. Okay, so exonuclease will remove the nucleotide from the 5' end or from the 3' end of the DNA molecule, but only one strand of DNA will be clipped by the exonuclease enzyme. Clear? Second one, endonuclease will clip the DNA duplex at any point within, in between. Alright, endo means inside, so within any DNA molecule it will clip the segments of DNA, the bases of DNA, but not at the end. They clip only one DNA strand. So you can see over here, it is cleaving the DNA. Endonuclease is cleaving the DNA within any base sequences within the DNA molecule. Within means inside, not at the end. Like in exonuclease, you are seeing that the nucleotides are being cleaved at the end, either the 5' end or in the 3' end, but the endonuclease will clip within, the inside of the DNA molecule. Clear this one? So endo means inside. Okay, so it will clip the DNA molecules within the base sequences, not at the end of the DNA molecule, but within the DNA molecule. Then comes the third one, which is the most important one, the restriction endonuclease. Okay, that will clip the both strands of DNA molecule. Listen very carefully. Exonuclease, endonuclease, they will just clip one strand of DNA molecule. But restriction endonuclease will clip both the strands of DNA molecule. Clear? This is most important thing. Okay, you have to remember, it will clip both the strands of DNA molecule. Then next one, this restriction endonuclease, all right, will clip the DNA molecule at a specific sequence of nitrogenous bases. That means a specific sequence of nitrogenous bases will only be recognized by the restriction endonuclease. All right, it will not recognize any type of base sequences, but to recognize, to clip the DNA molecule, it has to recognize a specific nitrogenous base sequences, and these specific sequences of nitrogenous bases which are recognized by the restriction endonuclease enzymes are known as recognition sequences or recognition sites or restriction sites or the target sites. Okay, so it will recognize specific sequences which are called recognition sequences, recognition sites, restriction sites or target sites, and these sites have got which sequences? They have got palindromic sequences of nitrogenous bases. We will discuss what they mean by palindromic sequence also. Okay, and there are specific restriction endonuclease enzymes for specific palindromic sequences. Clear? That means all the restriction enzymes, they don't have the common palindromic sequences, but for each palindromic sequences we have got specific restriction endonuclease enzyme. So for one palindromic sequences we have got one restriction endonuclease enzyme. For example, restriction endonuclease 1, EcoR1. Okay, that's just the example of restriction enzyme EcoR1. So this EcoR1, the restriction endonuclease enzyme, will recognize the palindromic sequences GAATTC. Now what does this GAATTC means? G means guanine, A means adenine, T means thymine, and C means cytosine. Okay, so this EcoR1 will recognize the palindromic sequences having the nitrogenous bases GAATTC in both the strands of DNA, that is from the 5' to 3'. If you remember, alright, DNA is a double-stranded molecule. Okay, double-stranded molecule. So suppose this is the two strands of DNA. Okay, so the polarity of DNA will also be just opposite. If one of the strands will run from if one of the strands will run from 5' to 3', then the other strands will be just opposite. It will be here 5', and here it will be 3'. Clear? So this is called the anti-parallel polarity of DNA molecule. All these things we have learned already in class 10 also, in 11 also. Okay, and we'll learn in details when we go to the unit number 2. Clear? So these sequences are present in the DNA molecules, and GAATTC palindromic sequences are recognized by the enzyme EcoR1. Now what do you mean by palindromic sequences? Actually, palindromic sequences, okay, before that look over here. As you can say GAATTC, here you can see polarity of DNA strands 5' to 3', and in the other strand polarity 5' to 3'. Base sequences you can see over here GAATTC. Okay, so this particular polarity or base sequences are recognized by the enzyme EcoR1. Clear? Okay, so what do you mean by palindromic sequences? So the term palindromes refers to the group of letters that form the same words when read both forward and backward. For example, rotator. So just read from here. Okay, read in this direction. ROTATOR, rotator. So when we are going from forward in 4 directions, all right, it's giving the word rotator. Now just come backward in the direction. ROTATOR. So such sequences are called palindrome. Here also MADAM, madam. Here also you can just see MADAM, madam. This is called palindrome. Okay, RADAR, radar. Or can come this side, RADAR, radar. Then Malayalam, MALAYALAM, Malayalam. Now go backward, MALAYALAM, Malayalam. So these are called palindrome. Now in terms of DNA, what do you mean by palindromic sequences? Okay, so in terms of DNA, palindromes are the base pair sequences that read the same in both the DNA strands. That means when we are reading the sequences from 5' to 3', you can see over here, when we are reading the sequence from the 5' to 3' directions in the forward directions, and again when we read the strands backward, again in this way, it has to be the same, right? So when we go from 5' to 3' direction, the nitrogenous base sequences you can see over here, it's GAATTC, right? Now let's come backward from 5' to 3'. Look at the sequences. This one is G, okay, because there's cytosine, there is guanine, GC, AT, GC. So this is G. Clear? So GAATTC. So that means these sequences are being read in the same way when we go from 5' to 3' directions, and again backward from 5' to 3' direction. So these such sequences are called palindromic sequences, and such sequences are being recognized by the restriction endonuclease enzyme. And the GAATTC is the palindromic sequences for which enzyme? EcoR1. Clear? So those sequences will be recognized by the restriction endonuclease enzymes and will clip it. Alright, restriction endonuclease will recognize these palindromic sequences GAATTC in both these strands and will clip it and will just make a cut over there. So can you see the arrow over here? This one. So I'll just make it clear. Okay, so it will be easier for you. So it has been recognized. Now after recognizing it, it will clip it. So can you see this green arrow? So the enzyme is cleaving the DNA molecules. Okay, now it is making a cut over there. So here you can see, after making the cut over here, in the first one 5' to 3', you can see over here GAATTC is there, right? But over here you can see that the sticky ends are being produced, TGC, TTA. So it has been removed, it has been cut, and ends are produced which are called sticky ends. So this we'll just see in details when we go through the next one, okay, to the next topic. It will be easier for you, but just for now remember the sequences GAATTC and GAATTC, which are being read in the same way when you go from 5' to 3', and again from 5' to 3'. Okay, so let's see now how this enzyme will come into actions. So first of all, let's see the nomenclature, the naming of restriction endonuclease. So restriction endonuclease are named by using the first letter of the name of genus. So whenever we are going to name the restriction endonuclease enzyme, the first letter, it comes from the genus. The first letter of the name of the enzyme comes from the genus and it is written in capital. Okay, then it is followed by the first 2 letters of the species. So first letter in naming the enzyme is taken from the genus, it's always in capital. It will followed by two small letters. It's coming from the species, okay, from which this enzyme has been isolated, and all the three letters are written in italics. For example, we are saying that EcoR1, right? EcoR1 is the name of the restriction endonuclease. So EcoR1. So the word, alphabet E is coming from the genus, right? According to this rule over here, first letter comes from the genus. So E is coming from the genus Escherichia. Can you see over here? E is coming from the genus Escherichia. It has to be in capital. Then it's followed by the two small letters which comes from the species from which it has been isolated. So CO, can you see over here? Coli, Escherichia coli. CO is coming from the name of the species. So Eco. So E is coming from the genus, written in capital. CO is coming from the species. Okay. Then R means the subtypes or the strains of E. coli. Strain means subtype. Okay, subtype of or the strain of E. coli. And the Roman number 1 indicates the order in which the enzymes were first isolated. Okay, so EcoR1. E is coming from the genus, CO is coming from the species, R is coming from the strain or the subtype of E. coli, and 1 is coming from the order in which the enzyme has been isolated from the bacterial strain. Okay, so you can see over here the derivation of the name EcoR1. E, source Escherichia, generic name, genus. CO means coli, coming from the species name. R means the strain, R1Y3 means the strain of bacterium, and Roman number 1, alright, Roman number 1 indicates the order in which the bacterium has been isolated. Okay, and for naming of these enzymes, for study of this enzyme EcoR1, W. Arthur H. Smith and D. Nathan has received the Nobel Prize in 1978 for the discovery of the enzyme EcoR1. And EcoR1 is a restriction endonuclease enzyme which recognizes the palindromic sequences GAATTC. Okay. Now let's come to the functioning of the restriction endonuclease enzyme. So this enzyme will recognize a specific palindromic sequences in the foreign DNA and cut sugar phosphate backbone of both the strands of double helix at a specific point in the palindromic site. That means these enzymes will recognize the palindromic sequences in foreign DNA. After recognizing it, it will make a cut at the sugar phosphate backbone of both the strands of DNA molecule. Okay, so look over here. So first of all, we have got over here vector DNA. That will be easier for you, okay, to understand. Then we have got the foreign DNA having the gene of interest. So first thing is that the same enzyme needs to be utilized for making a cut in the vector DNA and in the foreign DNA. So suppose we are using the enzyme EcoR1, then therefore the palindromic sequences for EcoR1 is GAATTC. Okay, so you can see the sequence over here, GAATTC. Again over here GAATTC. So this is the palindromic sequence. Here also GAATTC and GAATTC. This is also a palindromic sequence. So these sequences are recognized by the restriction endonuclease enzyme. So after recognizing these base sequences, the restriction endonuclease enzyme will make a cut at the sugar phosphate backbone of both the strands of DNA molecule. Okay, so you can see the arrow over here. So this means that they are cleaving the base sequences of DNA at the palindromic sites at both the strands of DNA, in the foreign DNA also as well as in the vector DNA also. Now it's just cutting. So here you can see that CTTA is there, right? CTTA is there, but this portion AATTC and G has been removed, has been cut. Can you see? So this portion, now look over here. So this portion AATTC and G has been cut down by the help of the enzyme restriction endonuclease. That's why we are getting this type of structures. Clear? So you can say over here G, then C, T, T, A, A. So rest has been cut down by the restriction endonuclease after cleaving it. Same in the foreign DNA. It has been cleaved by the restriction endonuclease enzyme. Now the rest has to be removed. So you can see over here A, T, T, C and G, same one. Okay, same sequences will be removed from the foreign DNA also. Okay, so these sequences are being removed. Okay, so G was there, then we have got C, T, T, A, A, or we can say A, T, T, C. A, T, T, C also be removed from the foreign DNA also, and we are getting this type of structures after being recognized and being cut by the restriction endonuclease enzymes. Is it clear for you how the cut is being made? Okay, now after the cut, what will is that the restriction endonuclease enzymes will expose the single-stranded free ends from each fragments of DNA duplexes. So these are the exposed single strands of exposed DNA molecule. Okay, and these overhanging single-stranded free ends of DNA are called the sticky ends. So these ends are called the sticky ends. Okay, these ends are called the sticky ends after being cut by the restriction endonuclease enzyme. Okay, so now what we require? Now these sticky ends have the got the complementary ends of DNA fragment. Okay, these sticky ends, they have got the similar complementary ends of DNA fragments from the same source or from the different source, and usually they can join according to the base pairing rule. They can usually join. So how the joining is possible? This is most important thing. Okay, how are they able to join with one another? Because we are saying that both the sticky ends have the complementary base, and if the base sequences are complementary, then they can usually pair up, and those single-stranded DNA can again pair up to form a double-stranded DNA. That is called denaturation and renaturation of DNA. Am I right? So now the sticky ends have been produced after being cut by the restriction endonuclease enzyme, and those strands have got the complementary ends of DNA fragments. So now these two strands sticky ends have to be joined. They have to be ligated. Okay, so these two sticky ends, one from the vector DNA, one from the foreign DNA, now they have to be ligated, they have to be joined, and for joining require the enzymes, right? One was restriction enzymes that was utilized to cleave the DNA and to make a cut. Then we require enzyme. First one, to join it, we require an enzyme called DNA ligase, because these enzymes join or seal the gap between the DNA fragment. So now, according to the base pairing rule, if the complementary bases are there, they will pair up, right? And they will form a double-stranded DNA molecule. So, sorry, look over here. T is there, right? A is there, adenine, thymine, adenine, adenine, thymine, adenine, thymine. So they will pair up according to complementary base pairing rule. So now the DNA fragment joins the sticky ends. So GC, then adenine thymine, adenine thymine, adenine thymine, adenine thymine, guanine, cytosine. So according to the base pairing rule, the sticky ends will be joined by the help of the enzyme DNA ligase, which will join or seal the gap between the DNA fragments. Okay, and this ligation is mainly possible due to the discovery of the enzyme DNA ligase. Then we require the enzyme DNA polymerase, which will help in the synthesis of complementary DNA strands on DNA template. Then we require the enzyme reverse transcriptase, which will help in the synthesis of complementary DNA strands from RNA template. Remember, whenever from RNA there will be formation of DNA, that is called reverse transcription, and for this one we'll require the enzyme called reverse transcriptase. Then we require the enzyme called alkaline phosphatase. They mainly remove the phosphate group from the 5' end of polynucleotide strand. Okay, so these are the few enzymes which are required in DNA technology also. Hope this is clear for you. So this particular DNA which is being produced is known as the recombinant DNA or rDNA. Is it clear for you? So first of all, remember one thing: the palindrome sequences which are being recognized by the enzymes restriction endonucleases, and how the cut is being made, and the sticky ends which are being produced are mainly joined with one another by the help of the enzyme DNA ligase. And for this we also require the enzyme called DNA polymerase, right? We also require the enzyme called reverse transcriptase. We require enzyme called alkaline phosphatase. Okay, and whenever the sticky ends will be joined by the help of the enzyme DNA ligase, a new DNA will be produced which is called recombinant DNA. Okay, so this is mainly the functioning of restriction endonuclease enzymes. Now in the book, go to the page number 584. You will get the table number 11.2, where you will find the list of a few restriction endonuclease enzymes, the recognition cleavage sequence, and the restriction products. Okay, so you will find it in the page number 584. So eco R1 as the restriction sites GAATTC. So this is mainly the complementary palindrome sequences. Okay, and can you remember the cleavage products, the stickeins which are being produced. Similarly, you have got the second enzyme, HIND2. HIND means Haemophilus influenzae. Okay, because H comes from Haemophilus genus, IND is coming from the species name influenzae. So by this way, just you have to remember the name of the resistant enzymes: eco R1, HIND2, okay, XMA2, then BAMH1, TAC1, and HA3. And similarly, you remember the restriction sites which are being recognized by the enzyme, and the cleavage products which are being produced after cleaving the DNA molecules at the specific base sequences by these enzymes. So today we have just finished about the first tool of recombinant DNA technology, that is the enzymes. Clear? So the next tool, that is vectors. Okay, then the competent host, then foreign DNA. All these things we will learn in the next class. So kindly go through the textbook and just go through the first tool of DNA technology, that is the enzymes. Okay, any doubt you can simply ask me, right, through the WhatsApp, or you can simply call me. Thank you and see you in next class.